We'll talk about bacteriophages, viroids, and prions. We'll discuss their structure and properties of the bacterial viruses, and we'll discuss the lytic and lysogenic cycles. We'll also discuss the difference between viroids and prions. We Hindus have been storing Ganga water in our homes for the purpose of cleaning and rituals for centuries. Antibacterial properties of the water of Ganga and Yamuna have been characterized by Hankin in 1896, and this was long before the genesis of the concept of bacteriophages, which was developed by de Herle in 1970. Hankin, in that year, 1896, reported the cure for diarrhea and cholera by using raw Ganga water. Also, in New Times, Notial in 2009 found the presence of self-purificatory properties in Ganga water due to its fluidity and presence of some unknown heat-liable peptides. These peptides were able to kill the pathogenic E. coli 0157-H7. These were called as your bacteriophages. Bacteriophages are highly host-specific, having the ability to infect and alter the composition of microbial community. In short, they are also called as phage. All bacteria are not infected by all phages, but most phages can infect a specific species of bacteria. Bacteriophage or phages are obligate intracellular parasites of the bacteria. They may use some or all of the host biosynthetic machinery. They are much smaller than the host, obviously, which is that the bacteria, and estimated to be widely distributed and ubiquitous. That is, they can be found in all reservoirs populated by bacterial hosts, from soil to intestines of animals. One of the densest natural source for phage and other viruses is the seawater. They have been used for over 60 years as an alternate to antibiotics. However, this much controversial area of research. Composition of bacteriophages. Different bacteriophages may contain different material, but they all contain nucleic acid and protein. Depending upon the phage, the nucleic acid can be either DNA or RNA, but it can never be both. The shape of the bacteriophages is also different from one species to another. Typical phage have a hollow head where the genetic material is stored and a tunnel tail. The tip of the tail have the ability to bind to specific surface receptors on the target bacteria. Once attached, the viral genetic material is then injected through the tail into the host cell. Here, it directs the production of progeny phages, often over 100 in half an hour of duration. These young phages burst from the host cell, killing it and infecting more bacterial cells. Here you can see the different bacteriophage families and their different type of shapes. On the left and top side, you will see double-stranded DNA is the nucleic acid which is inside these viruses. These viruses are non-enveloped. On the right-hand side, you can see double-stranded DNA, and these are the enveloped viruses. On the left-hand side bottom, you can see virus which have single-stranded DNA. In the center bottom, you can see virus Liviviridae, which has a single-stranded RNA, and you have on the right-hand side bottom Cystoviridae, which has double-stranded RNA. You can see that these different viruses have different type of shapes. In the center, you will always find nucleic acid, which is either DNA or RNA. Now let's talk about phage nucleic acid and phage protein content. The nucleic acid of phages often contain unusual or modified bases. These modified bases protects the phage nucleic acid from nucleases. The size of the nucleic acid varies depending upon the phage. The simplest phage have only enough nucleic acid to code for 3 to 5 average size gene products, while the more complex phages may code for up to 100 gene products or maybe more. When we talk about the phage protein content, the number and amount of different kinds of protein, they vary from phage to phage. The simplest phage have many copies of only one or two different proteins, while more complex phages may have many different kind of proteins. These proteins, they function in infection and to protect the nucleic acid from nucleases in the environment. For example, here you have your T4 phage. It is among the largest phages in the world. It is 200 nanometer long and 80 to 100 nanometer wide. The average range is 24 to 200 nanometer in length. The phage head can be of different types. That is, it can be icohedral, filamentous, or head tail in shape. Many, but not all phages have tails attached to the phage head. But this T4 does have. Here you can see on the top, that is the shape is going to vary, and this is your tail. The diagram is on the right hand side top. Complex phages have contractile sheath which contracts during an infection of the bacteria. At the end of the tail, there is presence of base plate and one or more tail fibers attached to it. These are involved in binding of the phage to the bacterial cell. What's happening here is, this is your head. In the head, inside you will have your nucleic acid. This is your tail part. The tail part contains of three parts. You have your contractile sheath in the center. You have your tail fibers which are going to recognize the bacterial surface, and the base plate which is going to attach to the bacterial surface and make the connection permanent. So what happens is, these tail fibers, they recognize the lipopolysaccharides on the surface of the microbes, on the outer membrane of the microbial cell. Once they attach to these outer membrane lipopolysaccharides or LPS, they confirm that this is the bacteria which is the host of the virus. Then this base plate is going to attach to the surface and cause a penetrating hole, and then the contractile sheath is going to contract down just like a spring which is inside a pen, and the genetic material, which is the DNA in this case, it is going to be injected into the cytoplasm of the bacterial cell. So let's take a look at these processes. Infection of the host cell has 4 steps. You have adsorption, you have irreversible attachment, you have sheath contraction, and you have finally nucleic acid injection. Let's look at adsorption. This is the first step, and it is mediated by the tail fibers or some analogous structures on the tail fibers which tail fibers void phages. So it is a reversible process. Tail fibers, they attach to specific receptors on the bacterial cell. These receptors, they vary from different type of bacteria and different type of host. And the host specificity of the phage is usually determined by the type of tail fibers that a phage has. Example of specific receptors includes the proteins on the outer surface of the bacteria, the lipopolysaccharides, the pili, and the lipoproteins. These receptors are on the bacteria for other purposes, but phage has evolved to use these receptors for infection. Irreversible attachment. The irreversible binding of phage to the bacterium is mediated by one or more of the components of the base plate. Phages lacking base plates have other way of becoming tightly bound to the bacterial cell. Sheath contraction. The irreversible binding of the phage to the bacterium results in the contraction of the sheath, and the hollow tail fiber is pushed through the bacterial envelope just like an injection. Some phages have enzymes that digest various components of the bacterial envelope and cause a hole and help in causing that hole in an easy way. Nucleic acid injection. When the phage has gotten through the bacterial envelope, the nucleic acid from the head passes through the hollow tail and enters the bacterial cell. Usually, the only phage component that actually enters the cell is the nucleic acid. The remainder of the phage remains on the outside of the bacterium. There are some exceptions to this rule. This is different from animal cell viruses, in which most of the virus particles usually get into the cell. The difference is probably due to the inability of the bacteria to engulf materials. This is your homework. You have to make short notes from this research article. This is, you go there on this web page, read it and make short notes, and this will give you insight about these bacteriophages and what are the things which are making the Ganges water so important. Now let's talk about the life cycle of these bacteriophages. Bacteriophages, just like other viruses, must infect a host cell in order to reproduce. The steps that make up the infection process are collectively called the life cycle of the phage. Some phages can only reproduce via a lytic life cycle. In this case, they burst and kill their host cells. Other phages can alternate between a lytic life cycle and a lysogenic life cycle, in which they don't kill the host cell, but instead the DNA is copied along with the host DNA each time the cell divides. We'll look into it in details in the next few slides. The lytic cycle and the lysogenic cycle. In the lytic cycle, the phage infects the bacterium, hijacks the bacterium to make lots of phages, and then kills the host cell by exploding it or by lysis. In the lysogenic cycle, the phage infects a bacterium and inserts its DNA into the bacterial chromosome. This allows the phage DNA, now called as a prophage, to be copied and passed on along with the cell's own DNA. The stages of the lytic cycles are: attachment is the first step. Proteins in the tail of the phage bind to a specific receptor, in this case a sugar transporter on the surface of the bacterial cell. Entry: the phage injects its DNA or RNA genome into the cytoplasm of the bacteria. The DNA copying and protein synthesis. The phage DNA is copied and phage genes are expressed to make proteins such as capsid proteins. Assembly of new phage. Capsid assembles from capsid proteins and are stuffed with DNA to make lots of new phage particles. Lysis. Late in the Lytic cycle, the phage expresses genes for proteins that poke holes in the plasma membrane and cell wall. The holes let water flow in, making the cell expand and burst like an overfilled water balloon. In this diagram, you can see the Lytic cycle. Here is the first step of attachment. This is your bacterial cell. This is your phage particle. The phage particle, with the help of contractile sheath and the base plate, it attaches to the surface, and once it has permanently attached itself, it injects its genetic material into the bacterium. Here, DNA replication and protein synthesis takes place. The DNA makes many copies of itself inside the cell using the host cellular machinery. It prepares the capsid proteins. Then it is going to assemble these viruses inside the bacterial cell. And then it is going to lyse the cell, causing the cell to burst open, allowing all the newly assembled phage particles to release into the environment. Once in the environment, each new phage particle can go and attach to new different bacterial cell. The stages of the lysogenic cycle. Most phages are either purely lytic, sometimes called as virulent phages, or capable of switching between lytic and lysogenic cycles, sometimes also called as temperate phages. The lysogenic cycle allows the phage to reproduce without killing its host. Attachment is the first step. Here, proteins in the tail of the phage bind to the specific receptors, again in this case sugar transporters, on the which are available on the outer surface of the bacterial cell. The entry of the phage is not the whole phage, but what it does is it injects its genetic material into the cytoplasm of the bacterial cell. DNA copying and protein synthesis. Once the phage DNA is inside the cell, it is not immediately copied or expressed to make proteins. Instead, it recombines with a particular region of the bacterial chromosome. That is, it is integrated into the host chromosome. Here you can see the diagram of the lysogenic cycle. The bacterium, or the bacterial cell, it gets attached by the phage particle. The phage injects its genetic material. This genetic material integrates itself into the bacteria's chromosome. This phage DNA, that is, it recombines with the bacterial chromosome. And thus it becomes prophage. Now, every time this cell is going to divide, it is going to make many cells with the prophage. And when the time will come, when the environmental factors will be in favor of the lytic phage, then these prophage genetic material is going to start with destruction of the cell and reproduction of virus particle. The integrated DNA, phage DNA, which is called as the prophage, is not active. That is, its genes aren't expressed as yet, and it doesn't drive production of new phages. However, each time the host cell will divide, the prophage is going to be copied along with the host DNA, getting a free ride. Under the right condition, the prophage can become active and come back out of the bacterial chromosome, triggering the remaining steps of the Lytic cycle: DNA copying and protein synthesis, phage assembly, and lysis. To lyse or not to lyse is the question. How does a phage decides whether to lyse or not to lyse? How does it decide what stage to enter, lytic stage or lysogenic stage? One important factor is the number of phages infecting the cell at once. Large number of co-infecting phages makes it more likely that the infection will use the lysogenic cycle. This strategy may help prevent the phages from wiping out their bacterial host. That is, by reducing down the attack, the phage to host ratio gets too high. What triggers a prophage to pop back out of the chromosome and enter the lytic cycle? At least in the laboratory, the DNA damaging agents like UV radiation and certain chemicals will trigger most prophages in a population to reactivate. However, a small fraction of the prophages in the population, they spontaneously go lytic even without these external cues. Bacteriophages versus antibiotics. Before antibiotics were discovered, there was considerable research on bacteriophages as a treatment for human bacterial diseases. Bacteriophages, they attack only their host bacteria and not human cells. So, they are potentially good candidate to treat bacterial diseases in humans. After antibiotics were discovered, the phage approach was largely abandoned in many parts of the world, particularly the English-speaking countries. However, phages continued to be used for medical purposes in a number of countries, including Russia, Georgia, and Poland. They are still used there today. There is increasing interest in bringing back the phage approach elsewhere as antibiotic resistant bacteria are becoming more and more of a problem. Research is still needed to see how safe and effective phages are, but who knows? One day your doctor might write you a prescription for a phage instead of penicillin. Viroides and prions. Viroides are the smallest infectious pathogens known. They are composed solely of a short strand of circular single-stranded RNA, which has around 300 nucleotides. It has no protein coating. They do not code for any protein, and they are inhabitants of higher plants, and most of these viroids, they cause diseases in higher plants. Prions, on the other hand, are misfolded proteins with the ability to transmit their misfolded shape onto normal variants of the same protein. The word prion derives from proteinaceous infectious particles. They characterize several fatal and transmissible neurodegenerative diseases in humans and many other animals. It is not known what causes the normal proteins to misfold, but the abnormal 3D structure is suspected of conferring infectious properties, collapsing nearby protein molecules into the same shape. These prions can bind to plants and be taken into the roots, where they may travel to the stems and leaves. Therefore, it is possible that prions excreted by deer or rat or human can pass on to other animals such as grazing cows, and through them even to humans. And especially if you have a prion infected plant and humans consume it, then the humans may also get infected. The diagram, in the image, you can see that on the surface of the plant, you can have contamination of the prions. This contamination can be through brain, urine, or feces, and there are chances that any plant eating animal is going to get infected. Also, if the soil contains prions, these prions can be uptaken by the plant through their roots and they can reach the leaves, and the animal consuming these leaves can get infected. Let's talk about the next topic: bacterial genetics and genetic engineering. Gene expression, we're going to talk about episomes and plasmids. We'll talk about genetic recombination, transformation, conjugation, and transduction, and genetically modified organisms. This is a topic in which we are only going to deal with few parts. We all know about the central dogma. Here, the DNA is is converted into RNA, and RNA makes the proteins. So this process by which information from gene is used in the synthesis of a functional gene product. These gene products are often proteins. Gene expression is generalized on central dogma, first formulated by Francis Crick in 1958. What you have to remember is that DNA replication is where DNA is made from DNA and the enzyme used is DNA polymerase. When you make RNA from DNA, this process is called as transcription. This process utilizes RNA polymerase as an enzyme. And RNA, they code for your proteins. This process is called as translation and it utilizes ribosomes. The central dogma is a two-step process. That is, it has transcription and translation, by which the information in gene flows into making a protein. The general transfer is described whenever you are making DNA from a DNA, it is called as DNA replication. When the genetic information in DNA is copied into making an mRNA, it is called as transcription. And when this information is utilized to make proteins, that is, when proteins are being synthesized by using the information from mRNA, this process is called as translation. There are certain special transfer described. Here, RNA can be copied from RNA. This is called as RNA replication. You can also have DNA being synthesized using an RNA template, which is called as reverse transcription. This is one of the methods by which reverse transcription polymerase chain reaction uses this technique in order to identify the number of viral particle in the COVID-19 patients. And sometimes it also happens, and it is very rare, but it does happen, that proteins can be synthesized directly from a DNA template without the use of mRNA. The two research articles at the bottom contains information on this particular information, this topic. Episomes and plasmids. The term episome was introduced by Jacob and Woolman in 1958. The purpose was to describe an accessory genetic element having the additional ability to integrate themselves into the chromosome semi-stably. That is, they could at some point dissociate from the chromosome and again replicate independently, or could even be totally eliminated, cured from the cell. We can see Ellie Woolman and Francisco Jacob in the images below. The episome concept was initially very useful in pointing out that two types of elements, these were namely the temperate bacteriophage lambda and F sex factor for conjugal fertility. They can both spend part of their life history integrated into the chromosome. The definition was also intended to include lysogenic bacteriophages that could either integrate into the chromosome and persist as prophages or replicate extrachromosomally and produce new burst of phage particles on cell lysis. The term plasmid was coined by Joshua Lydberg in 1952. They are physically separated from chromosomal DNA and they are named as extrachromosomal. They are most commonly found as small circular double-stranded DNA molecules in a bacteria. However, plasmids are sometimes present in archaea and eukaryotic organisms also. They can replicate independently. In nature, plasmids often carry genes that benefit the survival of the organism and confer selective advantages such as antibiotic resistance. Mr. Lederberg is on the left-hand side, bottom. He was the winner of 1958 Nobel Prize for his discovery of how bacteria transfer genes. This is your homework. You have to make short notes from the following article. You're going to go on this web page, biologydictionary.net/germ-theory/ and you're going to make short notes on this particular topic. Are the questions which you should be able to answer now? You have to go through the whole reading process and you will be able to answer these questions after going through the complete